Hi, I'm Harry Blake, an MRS student at the University of Portsmouth, and today I will be presenting my research regarding the Wenlock arthropods of Buttons and Quarry in Wales. So, as a result of its excellent exposure and vertical sequencing, Buttons and Quarry has attracted considerable research interest. Now, most of this research interest is focused around the graptolite fauna present, with very little attention paid to the particularly rare deepwater arthropods present. This is surprising considering that the ESCIE, or Early Shenwoodian Carbon Isotope Excursion, seen here on this modified chart from Sullivan et al. 2018, is present at Buttons Quarry, and its effects can be observed upon the arthropod fossils present. So here we can see the Shenwoodian age within the Wenlock epoch and the Silurian system in this chart, a partial chart from the International Committee of Stratigraphy. So here's a labelled map of Buttons and Quarry showing both its position in Wales and the various geological formations present, and this is from Lloydell and Large 2019. And here are a couple of pictures taken as part of my undergraduate degree on a field trip made to the site. So my project aims to do three things. First of all, investigate the paleoecology of the poorly understood deep water Euryptid present, as well as the very bizarre Ecumenical ostracods. Secondly, to constrain the controls on trilobite distribution throughout the material, as well as construct a range chart to aid in both of these. So in terms of methods used, specimens were first catalogued, revealing the presence of 1,914 arthropods. From there, a comprehensive literature study was performed, as well as SEM photos or scanning electron microscope photos taken in order to help with identifications and eventual production of a range chart, which will be used to correlate with other pieces of data. Pictured here are the material in its boxes, all arranged in stratigraphic height, or 12 boxes. So moving on to the results, of the 1,914 arthropods catalogued, 1,659 of those were catalogued as ostracods, and 30 of those have confidently been assigned to the family Ecumenidae based on the presence of a partial or a complete spine. Now, these ecumenids are concentrated around the upper area of the sample, specifically the upper Ricartinensis graptolite biozone, between the 8.5 metre to the 12.4 metre point, which marks the end of our samples. So here are some non-ecumenid ostracods, including on the left here a Bayerichian, as of yet unidentified, and on the left here Necogitis symmetrica. So moving on to the trilobite fossils present, of the original 1,914 arthropods, 254 of those were catalogued as trilobites, including two examples of Rhaphiphorus pavillus here, one of which might be a juvenile, and on the right here, a much larger Calamine blumenbach. So there are two distinct communities of trilobite present at Buttons Quarry. The lower Rhaphiphorus pavillus community, populated only by its namesake trilobite, which goes extinct after a brief period of bottom water anoxia, and is then replaced by a much more diverse community populated by Calamine blumenbachi, Exelaspis coronata, Ketneraspis crenata, and possible occurrences of Diphon barandi. Now, both Calamine blumenbachi and Rhaphiphorus pavillus are thought to have gone extinct locally as a result of bottom water anoxia. So moving on to the Euryptid fossils present, only one Euryptid fossil was managed to be recovered. This was assigned confidently to the family Carcinizomatidae based on its morphology, and more tentatively to the genus Carcinizoma. Now, both Gladwell 2005 and Lamsdale and Brady 2010 suggest that this Euryptid was a predatory Euryptid based on the presence of forward-facing eyes, though Gladwell goes on to elaborate that it probably had a nectobenthic, or swimming near the bottom lifestyle, based on its broad epithysoma or body. And here is the specimen in all its glory, with a single or possibly one or two podomeres present, and two distinct spines present, and finally a graptolite not to be confused as part of the present. And here is a reconstruction from Killis Vigrering 1958, where the distinct morphology of the chelicera can be observed, as well as its broad, unstreamlined epithysoma, which probably would have given it its nectobenthic lifestyle. So here are the people I like to acknowledge as part of this project, particularly my supervisor, Dr. David Lloydell, and Dr. Anthony Butcher and Richard Hing, who have both been very helpful with helping me complete this project so far. And any questions?